Welcome to Catch This. My name is Trudy Ray, and today I want to talk about immunity. You always hear people talking about B cells and T cells and antibody-mediated immunity and cellular immunity. But what does all of this mean? I will attempt to explain this terminology in this video. When we talk about antibodies and cell-mediated immunity, we are referring to adaptive immunity. Adaptive or acquired immunity initiates during a first exposure to a pathogen and protects from a second exposure to that same pathogen. This is in contrast to innate immunity, which is nonspecific and provides a broad response to any pathogen that has never been seen before. If you were to visualize adaptive immunity at a very high level, just to keep things simple, it helps to look at this graphic right here. During that first exposure, T helper cells sense the presence of one or more proteins known as antigens on the surface of the invading pathogen. And these T helper cells release a variety of signals known as cytokines that ultimately stimulate B cells to secrete antibodies to those antigens. However, antibodies only constitute half of the adaptive immune response. The other half, cell-mediated immunity, is just as important and at the very least results in activation of white blood cells that destroy ingested microbes and cytotoxic T cells that directly kill infected target cells. So just to reiterate, as part of the immune response that is often referred to as humoral immunity, because it happens in the humors or bodily fluids, B cells of your immune system produce antibodies. And as part of the immune response that is often referred to as cellular immunity, because it involves the combined action of cells, you have phagocytic cells that are capable of swallowing infected or diseased cells and cytotoxic T cells that can directly kill these cells. So let's look at this in the context of a specific infection, like SARS-CoV-2 infection, for example. Specifically, I want to ask the following question. Does a first infection with SARS-CoV-2 make a person immune to a second infection? We have been going back and forth on this topic throughout the pandemic. Some people say yes it does, and some are adamant that it does not. The authors of a review published in the Journal of General Virology earlier this year summarized some of the data obtained in regard to antibody responses to SARS-CoV-2 infection. They then compared these data to what is known about antibody responses to the 6 other coronaviruses that cause infection in humans. Yes, there are other coronaviruses. They include the 4 endemic seasonal human coronaviruses, NL63, 229E, HKU1, and OC43, as well as Middle East respiratory syndrome coronavirus, MERS-CoV, and SARS-CoV, the virus responsible for the 2002 to 2004 SARS epidemic. Remember that one? That is why the virus causing the present pandemic is called SARS-CoV-2. Most children produce antibodies to the 4 seasonal coronaviruses by the age of 6. However, this antibody immunity likely wanes over time because these viruses cause 22 to 25% of acute respiratory illness in adults. Even so, infection in adults results in low virus titers and usually only causes mild illness, suggesting that most infected individuals maintain at least some immune memory from their childhood. A study with human volunteers showed that 10 out of 15 adults inoculated with the 229E virus became infected, and 8 of these individuals developed clinical symptoms. There's a difference between becoming infected and becoming ill. Infection does not always guarantee disease, luckily for us. All 10 infected volunteers mounted antibodies that were neutralizing, meaning that they didn't just bind viral antigens, but inactivated the virus and prevented infection of new cells. These neutralizing antibodies peaked at 3 weeks after infection and dropped steadily until they reached baseline levels 1 year later. When previously infected subjects were intentionally infected with the same coronavirus at the 1 year mark, This is called a challenge study. 66% of them became infected, but none developed clinical symptoms. This suggests that there was sufficient immune memory to prevent disease. Similarly, research with MERS-CoV and SARS-CoV shows that a primary infection with these viruses results in total binding antibodies and neutralizing antibodies, and that both types of antibodies decrease to a minimal detectable level by 2 to 3 years after infection. Most of the SARS-CoV-2 antibody data obtained so far align with those observed with the other known coronaviruses, meaning that most infected individuals have detectable antibodies by 10 to 14 days after onset of symptoms. One group studying antibody responses in hospitalized people in China measured these responses using 3 different assays. The first assay detected total antibodies to the receptor binding domain of the SARS-CoV-2 spike protein. The second assay measured IgM to the same antigen. And the third assay measured IgG against the SARS-CoV-2 nuclear protein. IgM antibodies appear in the early stages of antibody-mediated immunity and typically bind very strongly to the antigens, to the extent that they often interact with other nonspecific antigens. IgG antibodies are made later, and they're a lot more specific than IgM. And they're the main antibodies because they provide the majority of antibody-based immunity against invading pathogens. The results showed that 93.1% of patients produced total antibodies, 82.7% produced IgM, and 64.7% produced IgG by about 11, 12, and 14 days after disease onset. More studies showing similar results are emerging. The gold standard for determining whether a first infection renders a subject immune to a second infection is the challenge trial, in which a previously infected person is intentionally reinfected with the same pathogen. And I just described a study like that done with seasonal coronaviruses, which only causes mild illness in most people. However, most experts consider human challenge trials for dangerous pathogens unethical. So for dangerous new pathogens like SARS-CoV-2, these experiments are usually done in non-human primates. One such study in rhesus macaques showed that all animals infected with SARS-CoV-2 were protected from a second infection, and that this protection was mediated by both antibody and T-cell responses. And this was demonstrated by mild clinical disease or no disease at all. Even though macaques are not people, their immune responses often parallel those of humans and can provide important insights into human immunity. Although most human and animal studies suggest that exposure to SARS-CoV-2 elicits a strong immune response, some people don't seem to produce antibodies after an infection. This anomaly may be due to the inaccuracy of existing antibody tests, which may lack sufficient specificity and sensitivity. There are also many different tests available that are not necessarily correlated against each other, and this may further contribute to inconsistencies. Currently available tests also don't distinguish between total binding antibodies and neutralizing antibodies, a difference that can substantially impact evaluations of immunity and conclusions about whether immunity has been achieved. Throughout the current pandemic, the public focus with regard to immunity has been on antibodies. This is partly because T cells are more difficult to measure than antibodies. Nonetheless, because a well-balanced immune response requires both antibodies and T cells, there is presently no reason to believe that infection with SARS-CoV-2 does not provide at least some level of immunity. Reports of SARS-CoV-2 reinfections that lead to severe disease the second time around are still rare. The ideal immune response is sterilizing, meaning that it completely protects against a new infection. However, considering the current fatality rate of SARS-CoV-2 in some people, even a low-level immune response that doesn't protect against infection but prevents serious disease is very welcome. In other words, immunity is not black and white, and even partial immunity could potentially save a life. Keeping all this in mind, we don't currently know if the vaccines that are being rolled out provide sterilizing immunity. There is a good chance that they only protect from disease, but not infection, so that a vaccinated person could still replicate virus at a low level and potentially transmit it to others. We just don't know at this point. And honestly, I can't think of a single existing licensed vaccine that does provide sterilizing immunity. But I may be wrong. That's it for today. My name is Trudy Ray. I'll see you next time on Catch This.